happens inside a human tissue. A human tissue contains millions of cells constantly making decisions. They change states, communicating with their neighbors, and respond to disease and treatment. In the young lab, we want to understand those decisions, what controls them, how they are organized in tissues, and eventually how we might predict or change them. We approach these questions by bringing computation and its experiments together. Our research has three closely connected directions. First, we decode the regulatory programs that create cell states and ask whether we can engineer those states, particularly dendritic cell and astrocyte, to improve anti-tumor immunity and neurodegenerative resilience. Second, we study cells in their native tissue environment, where they are, who their neighbors are, and how these spatial ecosystems influence disease and therapeutic response. Third, we develop computational and mathematical approaches to turn increasingly complex single-cell and spatial measurements into quantitative and eventually predictive models of biology. These aren't separate projects. They continuously inform one another. So what might you actually do during a rotation? If you're computationally oriented, you could work with single-cell or spatial multi-omic data to discover disease-associated cell states, decode regulatory programs, or develop new computational methods. If you're experimentally oriented, you could perturb candidate regulators, characterize dendritic cell states, or test how engineered immune programs affect function. But what excites me most happens in the middle. A computational discovery can generate an experimental hypothesis, and an experiment can reveal something that sends us back to the data and the model. You don't have to already know both sides. A rotation is an opportunity to learn. These ideas lead us into several very different biological systems. In cancer, we've identified dendritic cell and other immune or stromal states associated with immunotherapy response, and we're now asking what controls them and whether we can manipulate them in its spatial context. We use spatial technologies to understand how those cells organize within tumors and lymph nodes, while also developing computational methods to extract more information from these technologies. And in neurodegenerative disease, we use similar single-cell and multi-omic approaches to understand what particular cellular states and brain regions become vulnerable. The diseases are different, but many of the underlying questions are surprisingly similar. We also don't expect research to move in a perfectly straight line. A rotation usually starts with a concrete question that can be addressed in a few weeks. But as you generate results, I want you to ask why they look the way they do, challenge our original assumptions, and help decide what should come next. If the project goes into something exciting, there's plenty of room for students to take ownership and develop it in their own direction. Science is only part of what determines whether rotation is a good fit. You also spend a lot of time with the people. We are a relatively young and interdisciplinary lab. Some of us spend most of our time at a computer, some at the bench, and increasingly many projects require both. We discuss science frequently, celebrate each other's milestones, and learn from one another. Because we're still a relatively small group, students have direct interaction with me and meaningful opportunities to shape both their projects and the culture of the lab. And that brings me to probably the most important question. What am I looking for in a rotation student? You don't need to arrive knowing everything we do. I'm much more interested in whether you're genuinely curious, whether you enjoy thinking deeply about a problem, and whether you are willing to learn things outside your current comfort zone. If you're computational, we'll help you understand the biology behind your data. If you're experimental, we'll help you think quantitatively about what you are observing. And if you've already enjoying moving between both worlds, that's wonderful too. Most importantly, I want a rotation to be a useful scientific experience for you, even if you ultimately decide that another lab is the right home for your PhD. So, whether you're computational, experimental, or somewhere in between, if something you've seen here makes you curious, come talk with us. You don't need to know exactly what project you want to work on yet. That's part of what rotations are for. Come talk with me or anyone in the lab. We look forward to meeting you.